to yet another exciting session of Saturday series. So today, we would like to welcome Dr. Prasanna Ramani, who is the Associate Professor from Department of Sciences, Amrita Vidyapeetham. Sir has an ample amount of years of experience in education. Welcome, sir. Welcome, Prasanna, sir.
Thank you, sir.
Yeah, sure. So today we will be having a session on future of drug design and delivery, which is going to be an exciting session. So I would like to request the students, if you have any queries, you can just go to the chat box and you can type your questions. And the end of the session, sir will be trying to clear all your queries. So, and again, students are requested to just note down all the doubts they have. So it's because there will be a lot of important points that will be covered. So it will be helping you a lot in your future. So without any delay, I request Prasanna sir to take over the sessions.
Sure, sir. Thank you. Thank you so much for your introduction, and good evening, everyone. So I will just share my screen, and once you see my screen, maybe like we'll start the session. Can you please confirm whether my screen is visible?
Yes, sir, it is visible, sir. You can have it in PPT.
Yeah, sure.
Yes, sir. Now it's fine.
Can you see my cursor as well? Red dot which is moving left and right?
No, sir, I can't see that, sir.
Can you see like me underlining my name?
Yeah, I can see. Yes, sir. Yes.
Yeah, perfect. Okay, good. So thank you so much. So we'll start the session. So as been said, we are going to have some sort of introductory aspects about the future of drug design and development. So before me starting the lecture, like how I have planned the series, I mean the lecture talk, is the first half of my lecture, like you will understand like some introductory aspects about what is drug and some definitions and various drugs being employed in various disorders. And then I will take you to a zone where how a drug being designed currently, how people work on in coming up with a new drug, and probably like at the end we'll see like how the design, or maybe like how it is going to take it to the future, what's the future being involved in the drug design and development. That's a whole plan of my lecture. So so this is a slide, I guess, like you're seeing the slide number 2. So we'll try to talk about what is a drug. So we all know that, you know, at some point in our life, we would have taken some drug, either for a body pain or a headache, or probably like a fever, or probably like there are certain drugs which is being implied for cold, or maybe like say dysentery, various aspects. Probably like everyone would have come across some drug. So basically, you should know what is a drug and what is a poison and what is an inhibitor. So these things, this is a primary difference that you should understand very clearly. So any substance other than food, you know, food is a basic need of a living. You know, whatever you consume besides food, which is going to prevent or diagnose or treat, or which can relieve symptoms of a disease or an abnormal condition, we call that substance as a drug. So the drug will inevitably gives you a promising effect, or like a therapeutic effect. And besides the therapeutic effect or desired effect, there will be another effect which you call it as a side effects. So certain drugs will give side effects as a sleeping, maybe like some cough syrups, you might have heard about it. Consumption of cough syrup will make the person to sleep well, or probably like antihistamines, which are widely used for the common cold, or maybe like some allergic symptoms, right? Maybe like you might have heard about a tablet called cetirizine. That's quite commonly like available or prescribed by the doctor for allergic symptoms, which induces sleep. So if the side effect predominantly, if it is high, that's going to be a problem for the individuals. So if the therapeutic effect is less and if the undesired effect is more, then you call the substance as poison. So the drug what we say, it can be a chemical or a biological substance, or it can be a synthetic or a non-synthetic origin as well. So if you broadly look at the category of drugs, it can be from the natural origin or it can be from the synthetic source. So natural origin can be either it can be from the plant, or it can be from an animal, or it can be from an algae or a microbes, or the plants which are growing under the water. Anything can be, you can say, of natural source. Synthetic origin is like maybe like you can completely move on to the laboratory and where you start preparing molecules of various scaffolds. So there is another category called semi-synthetic. So basically you try understanding the molecule which is being available on the natural source, and then you try modifying it synthetically and then bring out a synthetic derivative of the natural source. You call it as a semi-synthetic source as well. So these are the main source we can find where the drugs are. Like, so how the drug acts is like, it acts on a target. So each drug will have its own target. So target is basically the enzyme or a protein or a DNA or an RNA. It can be of any sort. So it can go and sit and ensure that the undesired health issues are being taken into consideration. So I'm just going to take you some sort of a basic understanding of how the evolution of drug development is happening. I'm just putting a line like sort of a mismatch in the animation. So this is going to start with the identification of a disease. So you try understanding like what the disorder is on the person, and try to diagnose and through various diagnostic tools, and then understand like the protein which is involved in the particular disorder. The whole slide of what I'm presenting here is to give you an idea about starting from the disease identification until the product development, how it gets into products. That's how the slide I have prepared here. So a disease being identified, and then maybe like the protein responsible for that particular disorder, people will try to isolate. So which will approximately take to 2 to 5 years, and try to establish the structure of the protein through various spectroscopic techniques. So after that, they understand like, okay, this is a protein being responsible for the disorder. And then they try to understand which molecule can go and bind with the protein so that the protein will not undergo metabolism. So one thing should be very clear. Let's say, for example, the cell consists of DNA, and the DNA gets converted to RNA, and the RNA gets converted to protein. Of course, there are other small stuff all available in the cell, but let us not worry about other things like the Golgi bodies and other stuff. So basic unit is a DNA that gets converted to RNA, and the RNA gets converted to protein. So the protein which we are targeting on is over here. So when a person gets into a disease or a disorder, the DNA will undergo a mild mutation, and the mutation will produce a mutated RNA, and the mutated RNA will give out a different protein. That protein is the one we are trying to target here. So once you get the protein structure, what is being sent out of the mutated DNA, you will understand, okay, this protein belongs to this particular disorder, which comes out of the mutated DNA. So this protein undergoes metabolism, and then the metabolized product is used for cell division of this particular DNA, which is already been mutated. So that's a whole story about this particular protein evolution. So it is important to stop. Sorry. So it is important to stop the protein to undergo metabolism if it undergoes metabolism, it becomes a feed for successive cells, and the mutated cells will try multiplying more, and this disorder or the disease intensity will get more and more aggravated. So it is important to stop the cell multiplication. So we need to design a molecule which can go and sit on a particular site of the protein. I'll come back to this a little later. But however, like, it has to go and sit on a particular pocket where the metabolism is stopped completely or partially. So which is going to be like, almost like it takes almost 2 to 5 years to understand the molecule, which is going to be effectively going and sitting on the protein pockets to stop the metabolic activity. So after that, maybe like, this is going to be on a computer level. Maybe like you do some computational methods and come up with a molecule, and then you prepare the molecule in the laboratory and go with a preclinical testing on animals, which likely may have like almost like 1 or 3 years. And then you try doing the formulation of this molecule either in forms of a tablet or in the form of a syrup or in the form of injection or mode of delivery, whatever you want to do it. And then you start scaling up from the laboratory scale to the industry scale. So, and then what you do is like you just file an Indian patent. So whenever you just try come up with a molecule, you try filing a patent. So here it can be an Indian patent or maybe a US patent. Depends on the desired use of the drug. And then maybe like on the human trials, which is going to take 2 to 10 years, and then you file another NDA and then understand more about the drug and get the approval of FDA. FDA is like a Food and Drug Administration regulation body, which is going to take almost like 2 to 3 years, and then the drug is going to come to the market. So basically, like when you try identifying the disease and when it comes into the market, these are different stages that this evolution happens, which is a current trend. So obviously, like, the drug design and molecular modeling or virtual screening will be done at this stage. That's what I just indicated over here. Okay, so these are some of the points which I would like to bring in. On the drug or maybe the medicine part, what is existing here, maybe like quite known currently is the hand sanitizer. Everybody is using that. So what is that hand sanitizer? Like basically, like it has ethanol, or some sanitizer do have propanol or isopropanol. And there are certain sanitizers which is non-alcoholic, which the example being given here, or maybe like some other with more hydrophilic in nature, like say benzalkonium chloride or glycerol, which is most likely viscous in nature as well. So what basically it does is like, you take a sanitizer and then it try to destroy the cell wall of the virus or the bacteria, thereby destroying it completely. So when the cell wall being destroyed and it cannot replicate anything. So the cell wall protects everything. So you have a inside you have a viral genome. The cell, this is an envelope which has been covered here. It is a protein which is on the envelope. So when you use either of this, it disrupts the cell wall or the envelope portion, thereby destroying the entire unit, either it's a virus or the bacterial unit. Okay, this is another interesting point which I would like to highlight is like it's a cone-shaped snail. It's a toxin for therapeutic use. Of course, it is you can say it is a toxin. It's widely used as a muscle relaxant as well. So you can see the structure of the peptide unit. So you have a disulfide linkage over here, or it can have a triazole linkage over here. So the triazole linkage being introduced synthetically just to improve the stability of the molecule. So when you have a disulfide linkage, either with connecting with 2 amino acid, either with a long chain or a short chain. So under certain conditions, the disulfide bond breaks and forms a free a thiolic group. So you have SH, this bond breaks, and this also breaks. So it undergoes fission and then forms a free thiolic group, which gives a lot of instability to the molecule, and that the peptide is not going to be effective for the purpose of muscle relaxant. So instead, like the people have modified it with a triazole over here, so which has substantially improved the stability and the activity of the drug. Okay, so we have discussed this already in the previous slide. So you have a healthy cell, so you can have a DNA over here. So okay, so basically like this, I'm trying to give you an understanding about what is a cancer cell on, right? So in each cell you have a DNA, and the DNA produces RNA, and then that gets converted to a protein. So when the DNA gets damaged, or it is called as a mutated DNA, and that is going to be producing your protein, and that's going to give a wrong message to another cell, and which meant for cell proliferation. So that's going to be a problem when it gets accumulated in the various parts of the body. So and then you call it as, okay, lung cancer or a liver cancer or a prostate cancer or a breast cancer and so on. So what people do is, like, people have come up with a drug which can go and sit directly on the DNA. So you can target the troublemaking entity. So troublemaking entity is like either it can be a DNA, or it can be an RNA, or it can be a protein. So the drug that you are designing, or that you are designing the drug, so it can target the protein. So the protein targeting can be done where the protein gets locked by the molecule, and it's not going to undergo metabolic action, and the cell proliferation gets stopped. Or you can target on RNA. So the RNA is not going to produce the mutated protein, so thereby cell proliferation is maximum, it can be stopped. Or you can target the DNA itself straight, and which is not going to produce the corresponding RNA and its corresponding protein. So the targeting can be either with DNA, or it can be an RNA, or it can be a protein. But protein targeting is going to be like more easier than targeting a DNA, because DNA is a little smaller entity when you compare with the protein molecule, and it is a little complex as well, targeting a DNA specifically within the cell. Okay, coming back to this particular slide, what is a cancer cell? So I have explained it very clearly, like what is the cancer cell on. People have come up with a drug called Cis Platinum. So what the Cis Platinum does is, like, it try to lock the DNA, so directly on the DNA part, and it is not going to allow the mutated DNA to undergo the process of producing RNA, and thereby the mutation is very well controlled. Okay, so it's something like a DNA and chemo resistance. So let's say this is going to be your DNA. So the CGA are the nucleobase that you see, and this is the error part, right? So this error part meaning like this is a part where the mutation has happened. So there are DNA cutting agents are available. So where specifically you can go and cut that particular mutated part, and then you can patch up the DNA and ensure that it becomes something like, you ride a bicycle and then the bicycle gets punctured, or maybe you ride a two-wheeler, the two-wheeler gets punctured. So, and then you drag it to the mechanic shop, and then the mechanic checks out the puncture, understands where the puncture is, and then he seals the puncture out, and then he fills the air, and then the two-wheeler or either a cycle or a bike is ready to ride. So of course it is little easy, because to visualize cycle or a bike is pretty bigger entity. Doing on a smaller entity is pretty difficult, that you have to visualize. So people do work on fixing the mutation in the DNA where the error is. First is locating error is very difficult, and then cutting it on the right location is again a tough task. And then putting it back to correct it is again going to be a tough task again. So of course, there are many researchers across the world, people are working on this. So this is another drug we can say which does the job. Still, it is in evolution state, we can say. So it is called as volaparib, which traps the parab onto the DNA. The whole unit gets into the DNA and traps it, and then ensures that the repairing being taken in the right fashion. But still, people are working on how it can be implemented in a larger scale. Still, it is an evolution state, we can say. So there is another interesting chemistry, you call it as PNA. So the peptide nucleic acids, they are like considered to be the artificial DNAs. So if you have a mutated DNA on the strand, if the mutation being detected, you can prepare the artificial DNA and then cut the DNA and then pair it up with the synthetic one, and the cell gets into original form. So these are the various processes people are working across the globe. Okay, so cisplatin, coming to cisplatin. Cisplatin is a molecule, it looks like this. So it goes and sits on the DNA groove, and it is widely employed in various types of cancer like bladder or collateral or head or neck, lung, ovarian, and testicular cancer. It finds it very good application. Okay, so moving on next level on tuberculosis. Of course, TB symptoms are like coughing, weight loss, acute chest pain, often gets into a fatigue, fever, loss of appetite. So it should be like you should not check on your own whether you have any of the symptoms. So it should be a combination. Say I have listed like 6 here, so it could be like combination of 3+ we can say, or like 4+. So in order to be like all this should be present, maybe like if it is more than 3, with predominantly coughing and weight loss, with chest pain, we can say it is a pretty much symptom of tuberculosis. So the first line TB drugs are being listed here, like isozanate, like this is a drug which is being widely employed in the treatment of TB. Itabutanol, like another drug which is going to be another effective drug towards TB, and other big peptides and cyclic molecules have been also been employed on the TB resistant. Okay, so of course, this is another slide which I would like to bring in here. Like, of course, like at every moment, everyone will undergo some sort of a stress and anxiety. So there are certain hormones present in our body which triggers a lot with respect to stress and anxiety. Like this is the one which is responsible for the stress, we can say, corticosteroids. So these are drugs being employed for suppressing extraordinary stress or extraordinary anxiety on a person. So smaller stress, smaller anxiety, like it's not to worry about much. So you call it as an anti-depressant drug or anti-stress drug or anti-anxiety drug. Fluorocycline, like it's a drug which has been widely available in the market. Okay, so moving on next, the chemistry of cold medicines. Of course, everybody knows how the cold gets into the body. You have a blocked nose, and then you end up in getting a cough, and some people will end up in getting a headache or sore throat. So it depends on what the person has. There are different drugs being employed. With a blocked nose, like people, doctor prescribes various antihistamines as well, besides whatever being listed over here. A cough, of course, like you can see, either a dry cough or a cough with plum. The cold comes out of with mouth as well. So you got as expectorants or like suppressants will be given in case of cough. So headache, of course, you have a lot of painkillers like starting from ibuprofen or acetaminophen. Sore throat, antiseptics are being widely given, or maybe a combination of all will give you. So the point to highlight is like there are drugs, right? So you should look at the drugs which has a different structure, chemical structure, which acts on a particular disorder. That's what the highlight what I'm giving here. So moving on the next phase. Okay, I have we have discussed about the disorders and the drugs being employed. In general, like we said, like phase 1, 2, and all, like then phase 1 trial explicitly to say that it should be on the test drugs on the healthy individuals. So after the animal study is over and higher clinical trials are being over, so people do test the drugs on higher healthy individuals to understand like what happens on a normal conditions. That's going to be as a control, and we should understand the dosage, the safety, the side effects. In case a healthy person or if the disorder is slightly less, if the person is like maximum healthy, if the person consumes a drug, what are the side effects predominantly comes in. So healthy volunteers will be picked up and then these tests will be done. And in phase 2, that it will be like scaled up to a larger group of healthy individuals or volunteers preferably. And phase 3 is like a newer in a different geographical location, maybe like more into a colder climate or more in a hotter climate. People do a wider variety of tests, and then people go on to the FDA approval. There is another phase called phase 4. Of course, like there are situations where the drug over a period of time, even the phase 1, 2, and 3 will not get detected. Over a period of time, if the drug gives out predominant side effects, so that will be withdrawn from the market. So that is going to be a phase 4 of the process. So they try administer the drug in a bigger population, and then they understand that the drug efficacy is not that great, and then they pull out the drug from the market, and the drug will no longer be available in the market for the public use. That's going to be a phase 4. Okay, so now we have some sort of a broad idea about what is a drug and how drug acts on a system, different disorders, and how the different disorders are being taken care by different drug molecules, and what are different modules or maybe like different stages which are involved in the evolution of drug discovery process. So now what we are going to do is like we are trying to understand like how the mechanism of drug action is going to be. Probably like you might have studied this in your biology or probably in the chemistry as well. Maybe like enzyme lock and key mechanism, or maybe like enzyme ligand receptor mechanism. That's what I'm going to explain here. So the protein which we are targeting here is a protein which comes out of the DNA, which is already damaged, which produces unwanted RNA, and that unwanted RNA or the muted RNA produces mutated protein. So that protein is the one we are targeting here. So which you see here is the not the normal protein, is a disorder, like a not unwanted protein, or which is creating a trouble to the human being. So we have a ligands. Ligands are basically your drug molecule or drug-like molecule. We are trying to understand, like you keep different molecules, understand like which drug molecule is going to fit into this particular cavity. So in reality, you have a protein like this, right? So this is a protein like this. You have a pocket here. The pocket is like you have a cavity. Can you see the cavity here? So that cavity, this particular molecule is going to go and sit, which prevents the protein to undergo metabolism, or maybe like the protein should not break and come up with smaller units. So we will do a sort of a screening in a computer. You call it as an in silico methodology. So where you try to find which molecule will be the best fit for this particular disordered protein. So this cartoon gives you an idea. This particular ligand is not fitting because you have some sort of a gap over here, and again this is having a gap. Gap is basically like the molecule goes and interacts with the protein site through hydrogen bonding. So if the distance is larger, the hydrogen bonding will not be so effective. So the particular molecule may not be an effective agent to stop the protein from the further metabolism. So we can say that, okay, this particular blue part, the molecule, the best fit molecule is going to be the molecule which is going to get tightly bound to the protein pocket and through hydrogen bonding, and the protein is completely intact, and it is not going to undergo metabolism. So this is a cartoon which I'm showing here. So this protein is going to be like this, and this ligand is the ligand which is going to be here. You do a process called docking, and this is a blue part which sits on the whole protein on this cavity, and which is not going to allow to break the protein into smaller chunks. Because whenever there is a gap, that gap is a starting point of the protein to undergo cleavage. So we are trying to arrest that particular point. That's what this cartoon I'm trying to explain on. Okay, so during the drug discovery process, the first is the target identification. Target identification is basically the protein which is responsible for the particular disorder. That's the identification process. And then you have to validate the identification using various computational tools. Computational tools meaning like there are a lot of softwares and packages which will try to give you an idea whether the protein is of desired conformation, which is necessary for the molecule to undergo the docking process. When you do start doing it, you should be sure that the protein which I have is with a different, with the right conformation. So conformation meaning like, the orientation of the protein on a desired shape. And then lead identification is a molecule that you are coming up with, and then optimize it, and then go for preclinical phase, and then finally you call it as a drug discovery. Okay, so I'm going to take you to a route where the computational drug design can be done. So which is a part of your drug identification, target identification, target validation, lead identification, and lead optimization, partially on the lead optimization. So there is a program, or maybe like you use computational tools or the computer-aided drug design, where you come up with various programs. You try doing a evolution process and understand which will be the right protein conformation and which will be the right drug which can sit on a protein. So you call the process as molecular docking. You choose a protein structure, and then you end up in getting a molecule which can go and sit on a protein. You call it as a ligand. So in other words, you might have seen that sort of a receptor and the ligand, or maybe like a ligand and a host, or either it can be called as. So what basically you do is like you come up with a new term called hotspots. So what are hotspots? Basically, like you take a protein and you try understanding the portion in which the protein will try to undergo, it breaks up and gets into smaller chunks, which we are saying again and again, same thing. So the bond at which the protein is breaking, that spot we call it as a hotspot. So you try identifying the hotspot and then see that whether your molecule can go and sit on the hotspot, thereby forming a hydrogen bonding. So to form a hydrogen bonding, so it can be the molecule which can be a hydrogen donor, where the hydrogen can form an effective hydrogen bonding with the heteroatom present in the protein, or the molecule may have a heteroatom where it can comfortably attract the hydrogen from the protein, thereby forming a hydrogen bonding. So it can be a hydrogen acceptor or a hydrogen donor. So hydrogen acceptor is the one which accepts the hydrogen from the protein. Hydrogen donor is the one which lends hydrogen to form hydrogen bonding to the protein. So I'm just trying to understand, give you a small example. Like you have a toluene here, cyclohexa dienone here, and then you have aniline here. So you have toluene. If you look at the carbon doesn't have lone pairs of electron. Cyclohexadienone oxygen has 2 lone pairs. So obviously it can attract hydrogens to form hydrogen bonding. So similarly, you have aniline here. Aniline hydrogens can effectively form a hydrogen bonding with the protein structure, as well as the lone pair on the nitrogen can be donated to attract the hydrogen from the protein source as well. So it all depends. This cyclohexadienone can act as a hydrogen acceptor, whereas here this can act as a hydrogen donor as well as acceptor. So this is a raw I'm trying to say. This is not the final thing which you are trying to hear. So just to make you understandable, I'm trying to portray this particular message saying as hydrogen acceptor and hydrogen donor, but there are other programs and other terminologies. As you grow up, you will try to understand more. So if you have a structure like toluene, which is going to be unfit to be a drug likeness, because it doesn't have hydrogen donor or hydrogen acceptor. So if a molecule should go and form a hydrogen bonding, obviously it should have a heteroatom like this, or it should have like this, where the lone pair can participate in hydrogen bonding. Here it can be oxygen, nitrogen, or sulfur, or other halogens which can involve in hydrogen bonding. So I guess like the concept of hotspots and how the molecule is going to interact with the hotspots is clear. So probably like you can try typing your questions on the chat box. If anybody has any questions or any query or any curiosity questions, you are most welcome to type, and I'll be happy to answer your question. Anyway, moving forward. So what does it matter if you ask a question? It's very important because we want to stop the mutation at the protein level, or in the DNA level, or in the RNA level. Protein level is a little easier. That's what we are targeting on. So if you look at a molecule like this part of the molecule which has been shaded as a yellow part. So this part has been said as hydrophobic part because it is not going to dissolve in water. So you call it as hydrophobic part. And it is not going to have any hydrogen bonding as well. So this particular part where you have a nitrogen, so where you can see that hydrogen bond donor hotspot. So what do you mean by that? So you have a hydrogen here. That hydrogen can be a hydrogen donor with the protein, which has a heteroatom with a lone pair, and this hydrogen can form a hydrogen bonding. Similarly, this nitrogen has a double bond over here, and it has a lone pair, and this can be a hydrogen bond acceptor. This is what I'm trying to say in the last slide. So this is a hydrogen acceptor because it accepts the hydrogen from the protein through hydrogen bonding. So it donates the lone pair. So which donates the lone pair is hydrogen acceptor, and hydrogen bond donor is the one which donates the hydrogen to form hydrogen bonding. So this is a pretty important phenomenon and concept which is essential for the protein to get bound to the molecule which is supposed to be a drug. So when it goes and binds it, and it gives out other therapeutic effect which is a desired effect. So if it is going to be undesired effect, let's say poison, the poison goes and sits on the live enzyme and produces undesired sudden effect enormously high. As the person will undergo a drastic change in the system. Maybe it can be a food poisoning, one such example we can say. So probably a person will have food from unhygienic place, or maybe some sort of a food which doesn't suits the person, or the person may not have the required enzyme to digest that particular food. The food remain as undigested food, or it is a poisonic food. So that will try figuring out various reaction. So that's what I'm trying to say. The protein binding, or the molecule which is going to bind with the protein or the enzyme, is pretty much essential at this stage. Okay, so I guess like it's clear to you so far. So moving on next segment towards the futuristic part. So far, like we understood, like what is a drug and what is the functionality, disorders, how the mechanism is going to be, like what is the future. So that's the talk of today's evening. So if you ask me a question like this slide gives you an example. Let's say, for example, like a person has a headache, so or maybe a body pain. So what the person will do, like they will take, and then maybe like they go to the pharmacy, or the doctor prescribes a medicine, maybe like more commonly like Dolo 650, or maybe like some analgesic tablet. So Dolo 650 is like you are consuming the drug, say paracetamol 500. So you're consuming the drug which is already formulated on that particular weight. Maybe it's a 500 milligram of a drug, or a 650 milligram of a drug, or some cases, some steroids will also be given. Maybe like a person has a sudden cardiac arrest, or maybe like some cardiac problem is there. Like people do give steroids, maybe a 2 milligram or a 3 milligram, which is essential to overcome that particular disorder. So the point what here I'm emphasizing is each individual is unique. The dosage which I require may not be the same as the other person consumes, or the dosage which I consume may not be sufficient. So I keep on consuming it more till I recover completely. Sometimes this happens, or sometimes what happens, maybe I take a tablet and the headache goes off. So you may not be knowing like whether the headache goes off with 650 milligrams, or it can be 200 milligrams or 300 milligrams. Nobody knows. So it is essential to fix the dosage based on the requirement of the patient. You call the concept as personalized drug design. So as I said, if I consume, or if a person consumes 650 milligrams of paracetamol or 500 milligrams of paracetamol, the person may get cured from the headache or the body pain. Maybe 100 milligram may be sufficient. Rest 500 milligram is going to give an unwanted effect, or required as a side effect is going to be predominantly high. The desired effect is going to be there, but on a longer run, the undesired effect is going to stay on the body, which is not necessary for living. So in the future, on this particular part, now people are working on how to come up with the signature protein. The person who has a disorder will exhibit the protein. You try to quantify the protein, how much protein is responsible for that particular disorder. Accordingly, the dosage can be tuned. So for which, I'm not talking about headache or a body pain. This is a simple disorder. I'm talking about a higher level disorder like central nervous system, maybe like problem associated with central nervous system, or problems associated with cancer or Alzheimer or Parkinson, or maybe like certain localized disorder like elephantiasis, or maybe like rabies. Depends on the quantum of protein being generated. That signature protein can be quantified, and depends on that dosage can be given. That's how the new trend is going on. But however, like if I start telling more, definitely it's going to be complex at your age. So I hope like I have given some sort of an idea about the drug design and delivery part, and how the future is going to be. So the future is like I predict each individual is going to have some sort of a testing kit where the protein can be measured, and accordingly a dosage can be given. So that's going to be future. Maybe it can be something else as well. So right now across the world, most of the medicinal chemists or the biologists are working on how to identify or how to fine tune or get the information about what sort of protein, or quantifying the protein or unwanted protein, how much the patient is having. Accordingly, the drug can be designed and delivered. So that's a point of emphasis, I can say. So with this note, I come to an end of my lecture. So thank you so much for your patience listening. I hope you enjoyed the lecture by hearing my talk. So if you have any questions or any curiosity, thoughts, anything you want to ask, feel free to communicate on the chat box. I'll be happy to answer. Thank you so much. So do we have any questions? Do we have any questions?
Sir, actually, there is one question is asking for career opportunities.
Okay, so career opportunities.
I think we have two questions regarding that.
Yes, please.
Yeah. Career opportunity itself.
Career opportunities after they complete their plus two, or like in general, when you pursue this as a lifetime career.
Like in which industries he will be getting
okay, fine. So maybe like, whoever has asked, let me just explain it. Hope you will get some answer from my side. So basically, like, the drug, getting into a drug, many people are involved. So starting from a chemist, it's all going to start from a chemist. So if you want to pursue the career as a chemist, you are going to be the starting point of a drug discovery. So that's a starting initial stage. We can say, like, I'm a chemist. I can say that proudly, like, no, in my lab, we have developed a lot of lead molecules, and we have filed a lot of patents as well, which are active against the molecules which are active against various types of cancer. So a starting point is going to be a chemist. So once a chemist develops a lead molecule, okay, this molecule is going to be active for a particular disorder, and then the pharmacologist or the pharmacy people will come into role to understand whether the molecule can be given in the form of a tablet or a syrup or an injection, or it can be a cream. The mode of administration will be decided by the pharmacist. So if you want to pick up a career as a pharmacist, there you fit in. So after that, the patient goes to the doctor. Doctor diagnoses the patient and then prescribes a drug. So if you want to pick up the career as a doctor, you can pick up a career as a doctor as well. So there are like pretty much broader terms, like I've said. But if you look at the chemist opportunities, you can pick up a career with UG and PG as a chemistry, and then you have to do a higher research, which we call it as PhD or a doctoral research. And then you can continue your research in academia or probably in industry, where you can do this research and come up with a molecule where you can give it to the pharmacist, where they can start the process. I hope I answered your question. Else, please feel free to type it. I will be happy to answer your question.
This is all the questions about, sir. People, students are more interested to know the thing itself, career opportunity, placement, and those things.
Okay, so regarding placements, we can say, like, with respect to placements, if you complete your PG, as such Amrita is concerned, of course, most of people who pass out from Amrita, they prefer to go out to do PhD, either in India or like out of India. Most of them, like, that's how they pick up the career as. So besides, there are a small segment of people who start the career as an industrialist. After masters, they go for a company, and then they go and start working in R&D sector, either a synthetic chemist or a analytical chemist. So if you are in a synthetic chemist, you will be doing variety of reactions. If you are an analytical chemist, you will try to test or establish the structure of the molecule by doing some analytical process. So there is another segment called quality control or quality assurance, where they document the whole process, how the process is being involved. So we can have wider opportunities if you look at the chemist perspective. Of course, it is equal opportunity if you look at the pharmacist as well. Right. So of course, the diagnosis part also is equally important. So it's all depends on the interest of the student or the interest of you, how you want to pursue your future. So if you want to pick up a chemist, this is a pathway you can go on. Or if you are picking up as a pharmacist, that's a pathway you're going to go on. Or maybe like if you are picking up a doctor or a physician, be like that's a pathway you'll be going on. So I guess like I'm clear. So yes, just let me just type it. I will be happy to answer some specific questions put on into the explanation.
I think this is the only questions they have right now.
Okay.
So anyway, students, if you have any queries, you can just go to our website. We have our mail ID btech@amrita.edu. You can just give your questions. We will be answering all the questions later also. So thank you so much, sir. Thank you for the wonderful session. I think the students also really enjoyed it. Thank you. And again, students, we will be having next session next Saturday also. So I think you will be there in that. Again, Prasanna sir also will be doing a lot of sessions in future. Thank you, sir. Thank you so much.
Thank you.
All the best, students. Thank you.